everyone, and welcome to Miss Estruch Biology and this entire Topic 8 theory video. If you're looking for help creating a structured set of notes as you go through this video, I've got that for free in the description. Just click the link, download that workbook, and you're ready to go. If you're looking to really boost your grade though, have a look at the Biology Study Club. This is my monthly membership in which you get up to 4 live lessons per week from me, plus 2 other teachers who are highly experienced and also examiners. You get my flashcards included, my past paper analysis, my study tracker. You get discounts on all of my other resources, free access to my masterclasses and mock masterclasses. I mark your work, I set you mock papers, I mark your mock papers, plus so much more. Then join the Biology Study Club, linked in the description below. This is my monthly membership in which you get to join me to become your personal tutor, plus so much more to help boost your grade. But that is it for now. I hope you enjoy the video, and maybe I'll see you in the club very soon. Topic 8 for AQA A-level Biology, and we begin with looking at 3.8.1, alterations of the sequence of bases in a DNA can only alter the structure of proteins. So basically it's mutations, and a gene mutation is the alteration of a base in the sequence of bases for one gene. And this is probably going to occur during DNA replication, which is in S phase of interphase in the cell cycle. Gene mutations occur spontaneously, but the frequency at which they occur can be increased by exposure to mutagenic agents, and that's things like carcinogens and UV light or ionizing radiation. And because a mutation can change the DNA base sequence in a gene, that means it could result in a different sequence of amino acids in a polypeptide. And if that sequence of amino acid changes, then that can alter where the tertiary structure bonds will form, like the hydrogen ionic bonds, and that could result in it folding into a different 3D shape. And if a protein has a different 3D shape, it will have a different function and potentially make it a non-functioning protein. Alterations to genes like this can result in a mutation that could cause cancer, and cancer comes up a little bit later in this topic. So there are 6 different types of gene mutations, 2 of which you learn in topic 4, but now you learn addition. You've already covered deletion and substitution in topic 4, inversion, duplication and translocation of bases. So addition mutations is when one extra base is added to the DNA base sequence. So if we have a look at this original sequence here, we've added in an additional adenine nucleotide, and as a result we get a frame shift, meaning all of the bases downstream of that mutation shift one position, and they all shift one position to the right, and that causes a change in all of these codons, and therefore they will probably code for different amino acids, and that will result in a very different amino acid sequence and a non-functioning protein. Deletion is when one of the bases in your DNA base sequence is deleted. This also causes a frame shift, but this time all of the bases downstream of the mutation shift one position to the left. Again, you get different codons, therefore it's going to code for different amino acids and a very different amino acid sequence, and therefore a non-functioning protein. Substitution is when only one base is swapped, so it's just changed. You still have the same number of bases, and it will only affect the one codon that base is in. And because the genetic code is degenerate, meaning you have multiple codons coding for the same amino acid, even though this mutation has happened, sometimes it still results in the codon coding for the same amino acid, and therefore has no overall impact on the final protein. And if the mutation happens in the introns within a gene, which are the non-coding regions, again, it's not going to change the amino acids that it codes for. So here you can see an example. We've just changed the thymine nucleotide into an adenine nucleotide. Inversion is when a section of bases will detach from the DNA sequence, but when they rejoin, they are inverted, meaning they're back to front now. So this section of the code is back to front, and you get a completely different amino acid sequence. So we can see our original DNA here, and now we've got this section must have broken off, and when it rejoined, it's reversed itself, it's inverted. So that is going to code for 2 completely different amino acids now. Duplication is when one particular base is duplicated at least once. And because that means we're adding in bases, it's going to cause a frame shift to the right downstream of that mutation. So you end up with a different sequence of amino acids. So here's an example here. We've got the base thymine added twice. And then lastly, translocation is a section of bases on one chromosome detaching, and it attaches onto a different chromosome. This is a substantial alteration, and therefore it can cause significant impacts on gene expression, and therefore the phenotype could be very, very different. 3.8.2 then is gene expression. And we start with stem cells. And in multicellular organisms, there are a diverse range of specialized cells, but they all originated from undifferentiated stem cells. So a stem cell is an undifferentiated cell, meaning it hasn't become specialized yet, that can continually divide, and it can then go on to differentiate to become a specialized cell, as we can see here. And there's different types of stem cells based on their ability to differentiate. So we've got totipotent, pluripotent, multipotent, and unipotent. Totipotent stem cells can divide and produce any type of body cell. So these are able to differentiate into all cells. And they occur for a very, very limited time in early mammalian embryos, so shortly after the sperm and egg fertilise. Pluripotent are still found in embryos, so we can see here, you don't need to know this term blastocyst, but the embryo then develops into this blastocyst. The cells on the outside go to form the placenta, and the ones here in this inner mass are the pluripotent stem cells. So these pluripotent stem cells can differentiate into almost any type of cell, just not the placenta, because those are the ones on the outside that have already started to differentiate, but any other cell. And these are the ones that are typically used in research or to treat human disorders. So the stem cells could be used to regrow damaged cells in humans, such as to replace burnt skin, or even beta cells to try and treat type 1 diabetes, or neurons to treat Parkinson's disease. There are issues with this though, as sometimes the treatment doesn't work, or because stem cells can continually divide, they do continually divide and they can form tumours. There is also debate as to whether it is ethically moral, because in order to use it for treatment, you would have to make a therapeutic clone of the person you are trying to treat. And the embryo is then destroyed once the pluripotent stem cells are removed as well. So we also have multipotent and we have unipotent stem cells, which are found in mature mammals, meaning after the embryo stage. And they can divide to form a limited number of different cell types. Multipotent cells are, for example, bone marrow cells that we have as adults or even as children. When we say adults, we just mean you've been born in this instance. And they can differentiate into a limited number of cells, such as all of the blood cells. Whereas unipotent cells, uni meaning one, they can only differentiate into one type of cell. And an example of that is the cardiomyocytes. They are unipotent stem cells which regenerate into new heart muscle cells in small numbers. Now, induced pluripotent stem cells, this is something that has been produced scientifically from adult body cells. Somatic cells means body cells, using appropriate protein transcription factors to basically ensure that all genes can be transcribed again. And therefore, it makes it pluripotent in property, even though you might have taken a multipotent or a unipotent stem cell. And this overcomes some of the ethical issues because you're not then creating a clone embryo, doing therapeutic cloning, and you're then not destroying embryos. You're getting permission from an adult, taking some of their body cells and reverting them back into a pluripotent state. So the way this is done then, the iPS cells are created from the adult unipotent cells. These are altered in the lab to return them to that state of pluripotency. The genes that were switched off, which basically means those genes could not be transcribed, are now able to be transcribed again. And this is done using transcriptional factors. iPS cells are very similar to embryonic pluripotent stem cells, but as we said, it doesn't have those same ethical issues, such as the destruction of an embryo and not being able to get permission. Have shown a self-renewal property in that they can divide indefinitely to give limitless supplies, and for these reasons they could be used in medical treatment instead of embryonic stem cells. So if we have a look, then we've talked about this use of transcriptional factors in induced pluripotent stem cells, but we haven't gone through what that means. So you can control transcription. You can also control translation, but we're just going to focus on how you can control transcription first, meaning you can prevent it from happening or initiate it to happen. And this is actually what happens naturally when cells differentiate and become specialized, because every cell in the body has your entire genome, so all of the genes. But because of transcriptional factors and other things, but mainly transcriptional factors, some of those genes don't get transcribed, some of them do. And that's how you get cells that produce a particular range of proteins and become specialized. So in eukaryotes, transcription of target genes can be stimulated or inhibited using transcriptional factors. And that's what we're going to have a look at. So transcription of a gene will only occur when a molecule from the cytoplasm enters the nucleus and it binds specifically to the promoter region on DNA once it's in the nucleus. And those molecules that do that are proteins, and they're called transcriptional factors. Each one can bind to a different and particular base sequence of DNA. And this links back to topic 1, knowing that because transcriptional factors are proteins, that means they have a unique tertiary structure. So it gives it that unique 3D shape. And that means it will be able to bind to a sequence of bases complementary in shape. So remember that concept of tertiary structures and complementary shapes. So once it does bind, it can then stimulate RNA polymerase so that transcription begins. And that is how it's going to be initiating transcription. So we do then get an mRNA molecule that can then go on to be used in translation. So without the binding of that transcription factor, the gene is technically inactive because transcription is not happening, and therefore that protein isn't being made. And an applied example that you need to know about is estrogen. Now, estrogen is not the transcriptional factor, but it activates the transcriptional factor. Estrogen is a lipid-soluble hormone because it's a steroid hormone, and steroids are a type of lipid. So that lipid-soluble hormone, which is estrogen, can dissolve and move by simple diffusion from outside of a cell into the cytoplasm. And when it's in the cytoplasm, there's a transcription factor that has a receptor on it, which is complementary in shape to estrogen so that they can bind. And when it does bind, it causes a change in the tertiary structure of that transcription factor. And that change in the tertiary structure changes the DNA binding site so that it is now complementary in shape to a particular sequence of DNA bases. So that activated transcriptional factor moves into the nucleus. It will then bind to the promoter region of a target gene, which has the specific DNA base sequence that it's complementary to. And when it does that, it stimulates the RNA polymerase to bind, and transcription can occur. And then we get our mRNA molecule that can be used in translation. There's also another way that transcription can be controlled, and that is through epigenetics. Epigenetics, here is your definition, is the heritable change in gene function, but without changing the DNA base sequence. So 2 marks there for those 2 parts of the sentence. And these changes are caused by changes in the environment. So for example, stress and diet. And it can result in transcription being inhibited or initiated. And that is due to particular chemical tags that bind either to the DNA or to these histone proteins. So one of those chemicals is methyl groups. And methylation of DNA means methyl groups binding to DNA, which we can see here. These green circles are representing methyl groups binding to the DNA. And when those methyl groups are added to DNA, They attach to cytosine bases only. And this prevents transcription factors from being able to bind. And that is because when the methyl groups bind, it's going to attract the DNA and histone complex really tightly together. So it winds up like this. And as a result, because it's so tightly coiled, you've got genes which are tightly wound up, and therefore transcriptional factors can't reach them to bind. That means RNA polymerase can't be stimulated and transcription doesn't happen. The next chemical group is the acetyl groups. And if you have acetylation of histone proteins, that means the addition of acetyl groups, and they can bind to the histone proteins. Now, an acetyl group has a partial negative charge because it's got an oxygen in it. And the DNA also has this partial negative charge because it's got a phosphate group in it. And those two negatives will repel. And that's why the DNA and histone complex is not as tightly coiled. And as a result, the DNA is accessible. Transcriptional factors can bind to the promoter region on the DNA and stimulate RNA polymerase. So that means if you have decreased acetylation, meaning the acetyl groups are being removed from the histone proteins, that makes the histones more positive, and therefore they're going to attract to the DNA, and your DNA and histones are going to be more strongly associated, and it coils back up again. And again, that means that transcription factors can't bind. So that means that if you have increased methylation and decreased acetylation, that results in this tightly coiled complex of the DNA and histone proteins, and that will inhibit transcription. Whereas if you have methyl groups removed, so decreased methylation or increased acetylation, then you get this complex where it is not as tightly coiled and the genes might be exposed so that transcriptional factors can bind, and that initiates transcription. Now, you can also have control of translation after transcription has happened and mRNA has been made. And that is what RNA interference is, or RNAi for short. In eukaryotes and some prokaryotes, translation of the mRNA produced from the target genes can also be inhibited because of the RNA interference. So the mRNA molecule has already been transcribed, but that mRNA molecule gets destroyed. It basically gets cut up into small pieces, so it can't then attach to the ribosome and be used in translation. And small double-stranded RNA molecules are involved in this, typically known as siRNA, so small interfering RNA, or miRNA, microRNA. So here's our flow diagram of how protein synthesis typically happens. We've got transcription, that results in the production of mRNA. mRNA is then translated, and that results in the production of a polypeptide chain. So that is how protein synthesis occurs. Now, we've already talked about transcription could be prevented because, or initiated because of transcriptional factors and epigenetics. But here is where our siRNA or RNA interference has a role. So siRNA can cut up the mRNA to prevent translation, and therefore small interfering RNA results in RNA interference. So RNAi, the reason this is important is it's regulating gene expression, controlling development, and also it can fight viral infections and potential therapeutic uses for this could be silencing harmful genes and disease. So that's when it's going to be naturally occurring, and it could also be used in medicine in this way. The way this works then is in terms of siRNA, which we can see here. siRNA is produced from double-stranded RNA, and it splits into a single strand. It then is going to bind to a protein to make a complex, and one strand of that siRNA will bind to a complementary sequence of mRNA that's just been transcribed. And once it's bound to the siRNA in that protein complex, that protein is actually an enzyme that can cut up the mRNA. That is the level of detail that you need to know. I know it might sound a little bit vague, and that's because it is, but that's the extent that you need to know. It quite often comes up as application questions where they then give you a little bit more information. An alternative is miRNA, which was the microRNA. And miRNA is not fully complementary to its target mRNA molecule. So instead it binds to multiple mRNA molecules. And that often causes the blocking so that the mRNA can't attach to the ribosome, or it can also cause mRNA degradation as well. But either way, it results in translation not happening. So this then follows on to cancer. And cancer can result from mutations in genes that are responsible for controlling and regulating mitosis, as well as we're going to have a look at the role of epigenetics as well. But first of all, if these genes do get a mutation and a non-functioning protein is made, then that means mitosis is not regulated and you get uncontrolled mitosis or cell division, and that results in the creation of a tumour. Not all tumours are cancerous. They can be benign or malignant. So benign tumours, these are not cancerous. They grow, or they can grow very large, but just at much, much slower rates. And they're non-cancerous because they produce this adhesive molecule which sticks all of those cells together in a particular tissue, and they're often surrounded by a capsule. So that means that they're much easier to remove by surgery because you can remove the entire tumour, all the cells, because they're encapsulated. And also because it's in that capsule, it's localised, it's not going to break off and spread to other tissues. So it's often not life-threatening. However, that does depend on the location of the tumour because it will continue to grow and the treatment is surgery to remove it. So if it's in a location of the body where surgery would be difficult to remove it, such as in the brain, it could still be life-threatening. Malignant tumours are cancerous and they can grow large and they can grow large very rapidly. The cell nucleus becomes large and the cell can then become unspecialised again. They don't produce that sticky adhesive layer and they also don't have a capsule around them, which means that these tumours are able to metastasise. And what that means is cells from the tumour can break off and get into the bloodstream or the lymphatic system and therefore move around the body and then lodge in new tissues to make secondary tumours. The tumour, as I said, isn't encapsulated, and that also means it can grow out projections and reach blood supply. And as soon as it gets a blood supply, it's got lots of glucose and oxygen for respiration, which is why it can divide so rapidly. It can be life-threatening, and as well as trying to remove tumours, you can also have treatments such as radiotherapy and chemotherapy. But it's much harder to successfully remove the tumours because they're not encapsulated. It's very hard to tell if every single cancerous cell has been removed in surgery. And that's why it's often, if you do have surgery, accompanied with radiotherapy and chemotherapy to potentially kill any cells that might have been left behind in the surgery. So tumour development can be due to gene mutation in either a tumour suppressor gene or an oncogene, which are two genes involved in controlling mitosis. Or it could also be linked to epigenetics if there's abnormal methylation of a tumour suppressor gene or oncogene, or increased estrogen concentrations linked to the idea of transcriptional factors. So let's just go through what these two genes are. Oncogenes are the mutated version of a proto-oncogene. And a proto-oncogene creates a protein involved in initiating DNA replication and mitosis when the body does need new cells. An oncogene mutation results in that gene permanently being activated. So you are going to have the protein constantly being produced to initiate DNA replication for cell division, even if new cells aren't being made, and that's why a tumour forms. Tumour suppressor genes produce proteins or code for proteins that slow down cell division and cause cell death. So when you no longer need new cells, it make the protein, and also cell death if a copying error is detected, so that cell needs to be destroyed. Now, if a mutation results in the tumour suppressor gene, that means you won't make that protein anymore, and therefore cell division isn't going to slow down. And also potential mutated cells aren't identified and destroyed. And again, that is why it can result in a tumour forming. So if you have abnormal methylation, which links back to epigenetics, that means that this methylation could result in a gene being inactivated. And if that's the tumor suppressor gene becoming hypermethylated, meaning lots of methyl groups are binding to that DNA, that's going to cause it to tightly coil. The transcriptional factor can't reach the gene, the tumor suppressor gene, and the tumor suppressor gene is not transcribed. So it means you don't get that protein that's needed to control the cell cycle. Or the opposite could occur in an oncogene, as they might be hypomethylated, meaning you don't have enough methyl groups binding, and as a result, the gene is permanently being transcribed. Now, the link to estrogen is after menopause in females, estrogen is no longer produced by the ovaries. While the menstrual cycle is active before menopause, the ovaries is the main place that estrogen is produced. But after the menopause, the ovaries stop making estrogen, and instead it's mainly produced by fat cells in the breast tissue. So in postmenopausal women, this local estrogen production, constantly being exposed to the breast cells, increases the risk of breast cancer. And that goes back to what we looked at in transcriptional factors, because estrogen can bind to receptor proteins on transcriptional factors to activate the transcriptional factor. And that transcriptional factor can then bind and initiate transcription. So if estrogen stimulates transcription of a proto-oncogene, it can cause that gene to be constantly transcribed, and therefore you're going to be making that protein all the time that stimulates cell division, and that can contribute to a tumour forming in the breast tissue, because that is where this estrogen is being produced. And this can result in a positive feedback loop, because if you're making more tumour cells, which are breast tissue cells that are making estrogen, your concentration of estrogen is now even higher in the breast tissue, so this process is going to happen even more. So that is why it can then happen very, very rapidly. Next, we move on to 3.8.3, using genome projects. So genome sequencing involves working out the entire base sequence of an organism's DNA. And projects have successfully sequenced the genome of many organisms, including humans. So this is just showing us the progress through that. It's a bit blurry, but many organisms have had theirs sequenced, and for humans it was finally completed, the Human Genome Project, in 2003, but it had been ongoing for many, many years. Scientists across the world all contributing to sequence our DNA. So the applications of this, first one, simpler organisms. Simpler organisms, for example, bacteria. Genome sequencing is very useful because it can be used directly to predict the proteome, which is all the proteins an organism can make in their cell. And that's possible because simple organisms like bacteria don't have non-coding DNA. They don't have any introns, they don't have other non-coding sequences, and also they don't have regulatory genes as well. So that means the relationship between the genome and the proteome is very straightforward, and it's very easy to predict all the proteins they'll potentially make. And this has been useful in identifying potential antigens that bacteria could make, and therefore making vaccines against those particular bacteria. In complex organisms, for example humans, predicting the proteome after working out our genome is more difficult, and that is because we've got large amounts of non-coding DNA, and also we have regulatory genes that affect gene expression. So that means that knowing the genome sequence doesn't always directly reveal which proteins will be produced. Now, there are constant advances in sequencing technologies, which is why the spec doesn't get you to learn any, because they probably become outdated within a year. So that's why instead you just need to have this awareness that sequencing methods have become increasingly rapid, far cheaper and cost effective, and automated. And because of that, large-scale genome projects have enabled researchers to sequence even more organisms in less time with greater accuracy. So we've gained even more information. We then move on to 3.8.4, gene technologies, and the title goes on, but it's basically what we all call gene technologies. So gene technologies involves recombinant DNA. And recombinant DNA means you're transferring fragments of DNA from one organism, or one species, into another organism's DNA or another species' DNA. The organism that receives the DNA is called a transgenic organism or genetically modified. The reason it's possible links back to topic 4. The genetic code is universal, which means the same codons on mRNA will code for the same amino acids in all organisms. And that's why you can take the DNA from one species, put it into the DNA of a different species, and it will still code for exactly the same sequence of amino acids and make that protein. Now, as well as that, it also works because transcription and translation are universal as well. So that means genes from one species can be expressed in another. And that's because once you insert that DNA into the new species DNA, transcription will occur to make mRNA, and that mRNA will then be translated to make the functional protein in the host cell. So the ways that this has been used is producing insulin, for example, taking the human gene for insulin, putting it into bacterial DNA, and then bacteria will make the human protein for insulin. And that has been massively beneficial because bacteria can create large quantities very rapidly of insulin, which can be used to help treat diabetes. It's also useful to make transgenic crops with beneficial traits, for example, inserting genes so that they're naturally pest resistant, or even inserting gene function for research models to try and work out the role of genes in terms of helping with medical research. So how this is done then, I've split the rest of this topic into this flow diagram because from my experience teaching this, one of the challenges with the gene technologies topic is students learn all of the technology separately and don't see how it all links together, and therefore struggle when it gets to the challenging application questions when it is all linked together. So here it is and how it all links. One branch is genetic screening, and this is where you might screen to see if you have a particular gene. That leads to genetic counselling. Now this is done using gel electrophoresis, and it can result in personalised medicine. Genetic fingerprinting, that is how you can examine an individual's DNA based on the VNTRs, those variable number tandem repeats. And that is used in forensic science, medical diagnosis, paternity tests and more. And one of the stages in genetic fingerprinting is gel electrophoresis. Then we have over here how we actually make recombinant DNA. And the first step is if we want to put the DNA from one organism into another, we need to get that piece of DNA to start with. And that's what creating DNA fragments means, getting the gene of interest that you want to insert into a different species DNA. So here are the processes of how you can do that. Once you've then isolated that gene of interest, we then need to get lots of copies of it before we go on to try and insert it. And you can either get large numbers of copies in vitro, which means outside of the body, and that is using PCR, polymerase chain reaction, or you can do in vivo cloning, which means making large numbers of copies of that gene inside of a cell. And that's typically done inside a bacteria, and here's all the processes how. So that's a very brief overview. Let's go into it in more detail, starting with how we create these DNA fragments. So the combining of different organisms' DNA, which could enable scientists to manipulate and alter genes to improve their industrial processes, or we looked at medical treatments, always starts with you have to get your gene of interest, which means isolating the DNA fragment. And there's 3 methods: reverse transcription, restriction endonucleases, and the gene machine. So reverse transcription, this is using an enzyme. And this enzyme makes DNA copies from mRNA. The enzyme actually isn't written yet, but it is reverse transcriptase. So that enzyme is an enzyme that can make DNA copies from mRNA. And it naturally occurs in viruses. So you learn in topic 2 how HIV has the enzyme reverse transcriptase, and that can make DNA copies from mRNA. So the reverse of transcription. A cell that naturally produces the protein of interest gets selected. Then large amounts of mRNA for that protein should be in that cell and they can be isolated. You would then mix that mRNA that you've isolated with the enzyme reverse transcriptase. And what that will do is reverse transcriptase can join DNA nucleotides together, so via a condensation reaction, creating phosphodiester bonds, based on mixing together DNA nucleotides, exposing it to this mRNA, and the complementary bases will be attracted and aligned, and then reverse transcriptase can join to create that new DNA molecule. But because we'll have one mRNA sequence, and then you get your DNA sequence aligning and being created against that, you end up making a single-stranded DNA molecule. And we call this cDNA, complementary DNA, so complementary copy of our mRNA. So we then do have to make it double-stranded so that we can use it. And to make it double-stranded, we then need to make sure we've still got lots of DNA nucleotides present, and then we use the enzyme DNA polymerase. Now, the advantage of this method is that complementary DNA or cDNA won't contain any introns because mRNA doesn't have introns in it. They've already been spliced out. So that means we get a DNA copy with no introns in. And then we can see in this diagram, they then go to use PCR, so you get large numbers of copies of our cloned gene. Restriction endonucleases, this is another method using enzymes, and these are enzymes that can cut DNA. They naturally occur in bacteria, and bacteria have them as a defense mechanism. So if a virus was to inject their DNA into the bacteria, they naturally have restriction endonucleases to cut up viral DNA so that they can't be infected. And there are many different types of restriction endonucleases, and this links back to the idea of enzymes having a tertiary structure, which gives them a unique 3D shape, so their active site is complementary in shape to a particular sequence of DNA bases. So these restriction endonucleases, loads of different ones, so that they each have a different active site complementary to a different sequence of bases. They can then bind at that particular sequence, and they will break the phosphodiester bond at that point. Some enzymes cut at a location in a double strand to get a blunt end. What that means is it would cut directly down, so you get two straight lines, a blunt end. But most of the time, and in terms of use, it's more useful than they would use enzymes that cut at a jagged line. And that then gives you an overhang of bases here and an overhang of bases there. And that is known as sticky ends. And sticky ends are useful because that means that they can align opposite another fragment with an overhang, and it's much easier then to join them together because a complementary bases can align. And this is going to happen at these palindromic sequences. And that means the bases, the order of the bases is the same forwards as the order of the bases is backwards. That's what we mean by palindromic. So where these enzymes can bind and cut is known as the restriction site or recognition sequence. Those are the two terms that we use to describe where the enzyme can bind. That is based on the active site being complementary to a particular sequence of bases, and it creates these sticky ends. A G machine, this is when the DNA fragments can be created in a lab using a computerized machine. So first of all, the scientists will examine the protein of interest to identify what the amino acid sequence is for that protein. Then they can work backwards and work out what the mRNA sequence is, and therefore what the DNA sequence is. That DNA base sequence is then entered into the computer. Checks are made that you're not trying to make a protein that could potentially cause risk to human health, and other biosecurity checks, and that it's ethical to produce. Once those checks are in place, then the computer can create small sections of overlapping single strands of nucleotides, and these are called oligonucleotides. Those can then be joined to create the DNA for the entire gene. So once you've then created the entire gene, PCR is used to get large numbers of quantities and to make it double-stranded. So this process, the advantages are it's very quick, it's accurate, you can design it to be intron-free as well. And the reason we think intron-free is an advantage is prokaryotic cells don't naturally have introns. That means they don't have the cell machinery to splice introns out. So if we're going to be putting the gene into a bacteria, we need to remove the introns because they can't. So having it intron-free in the first place is a big advantage. Okay, so we've made our gene fragments. Now let's have a look at how you can clone them, meaning get large numbers of that gene of interest. First of all, with in vivo cloning. So we've already created the DNA fragment. Now we need to look at how we get that fragment into the vector and how we get that into the host cell. So the first thing though is adding a promoter and terminator region. So when we insert a gene into a host cell, so basically when we take one gene and put it into the DNA of another organism, it's not enough to include just the DNA base sequence that codes for the amino acids. For transcription to successfully occur, you also have to add a promoter region at the start of the gene. And the DNA sequence is inserted before the gene. And that acts as a binding site so that RNA polymerase and transcriptional factors have something to bind to so they can initiate transcription. The correct promoter must be used for the host organism though, so that the gene is expressed properly. Now at the very end of the gene, a DNA base sequence is added as well, and that's called the terminator region. And that will signal the end of transcription, and it ensures that RNA polymerase will detach and it stops transcribing at the correct point. So they are important then because without a promoter region, the gene wouldn't get transcribed. And without a terminator region, transcription might continue so that it then continues to transcribe unintended regions. And that would be wasteful of resources, and also it might disrupt the other genes. So basically these sequences, the promoter and terminator region, are essential for being able to produce functional mRNA when you do this gene technology. So how we get that DNA into a vector. But first of all, what is a vector? So a vector is something that will carry the DNA fragment that we've made into the host cell. That's what a vector is. And the most common vector that we use is plasmids, so those small loops of DNA that some bacteria have. So that's what we're going to be doing, getting our DNA into the plasmid first of all. And the way that we do that is the plasmid has to be cut open using a restriction endonuclease, but you have to use the same restriction endonuclease as the one that you use to cut out your DNA fragment. And the reason for that is that means your plasmid and your DNA fragment will have the same sticky ends. So that means they'll have complementary sequences overhanging that can align. And therefore, they are going to be able to align and join. Now, a few things just to give you extra detail that the mark scheme's more particular on now is you need to know the enzyme ligase is going to be joining the plasmid and the gene of interest together. But specifically what ligase does then is it's catalyzing the condensation reaction, forming the phosphodiester bonds between those nucleotides to join those two fragments together. So now we've got our gene of interest into the vector, the plasmid, we have to get that vector into the host cell, which is the cell that's going to make the protein, which in this case is a bacteria. And in order to do this, we need to make the host cell's cell membrane more permeable, so we're more likely to get the plasmid in. And the way that we make the cell membrane more permeable to increase the likelihood of the plasmid getting inside is using calcium ions and heat shock, which means sudden increase and sudden changes in temperature. So that's going to affect the fluidity of the cell membrane, so we should be more likely to get the plasmid in. So we've now cloned it, but what we have to do is make sure that before we grow that bacteria, we are only growing the bacteria that has definitely taken up the recombinant plasmid. And that's what we mean by identifying transformed cells. A transformed cell is a cell that has taken up the recombinant plasmid. Recombinant means recombined. It's made up of two different types of DNA. Now, not all of them will be recombined or transformed cells because, first of all, maybe that recombinant plasmid, even though we heat shocked the cell membrane, it still didn't get inside. Or maybe a plasmid did get inside, but it wasn't a recombinant one. The original plasmid that was cut open simply rejoined, so maybe that got inside, so we don't have the gene of interest. And sometimes that DNA fragment that we created actually connects back around to itself and makes its own little loop of DNA. And if that went in, then again, it wouldn't work. So we have to identify which bacteria have successfully taken up the recombinant plasmid. And that's done using something called marker genes. And marker genes are on the plasmid and used to identify which bacteria have taken up the plasmid. There's 3 marker genes that are typically used. Genes that are coding for antibiotic resistance, genes coding for the creation of proteins that fluoresce, so they go bright yellow or green, particularly under UV light, and genes coding for enzymes that can catalyze reactions causing colour changes. Now, you don't need to know the details of how that is done, but it's a common application question. So that's why I'm going through this. You aren't expected to remember this, but it comes up as application a lot. So that's why I'm going to go through this example with you. So I'm going to do it with antibiotic resistance. And we can see that here is the plasmid. And there are 2 genes for antibiotic resistance. This one here makes the bacteria resistant to the antibiotic tetracycline. And this gene here will code for a protein that makes the bacteria resistant to the antibiotic ampicillin. And here is the DNA fragment that we've just isolated. Now, what they would do is deliberately insert that DNA fragment into one of the marker genes. And in this case, they've put it in the middle of the tetracycline gene. And what that does is our DNA fragment can be transcribed and make a protein, but the tetracycline one has been split in half now, so that is not going to be able to be transcribed and translated to make the protein that makes it resistant to tetracycline. So any bacteria that takes up this plasmid will not be resistant to tetracycline anymore, but the resistance to ampicillin gene is still intact, so it will be resistant to ampicillin. So to work out which bacteria have taken up the plasmid, the recombinant plasmid, you would grow your bacteria on agar, and we can see all the colonies here that have grown, and we've labelled them. You would then use a sterile velvet block to make an imprint of this master plate and copy all of those colonies onto a second agar plate. And this time the agar plate has got agar with ampicillin antibiotic dissolved within it. And we can see that now some of the colonies have died and only some can grow. They would then take another imprint and this time grow those colonies on the agar plate that's got tetracycline dissolved within it. And now we can see only these 3 colonies can survive. And from this, we can work out which colonies contain the recombinant plasmid, because our bacteria that have the recombinant plasmid are resistant to ampicillin, because that gene's intact. So that means it has to grow on this plate, but it's not resistant to tetracycline anymore, which means tetracycline will kill it. So that means it won't be present on this plate. And that is only colony E and G, because they're on this one, they're not present on this one. So that tells us that E and G are the only colonies of bacteria that have that recombinant plasmid. So those are the ones that you should then remove and grow en masse. Next then is how you could do this, but using PCR. So PCR is our other way to get large quantities of the DNA fragment of interest. And vitro means not inside of a living cell. It's in a lab. So you can use the polymerase chain reaction to do this. It's an automated machine, so you get large quantities very rapidly. And PCR is going to be used probably multiple times per day in all genetics labs because it's so quick and you always want large numbers of copies of everything so you can do multiple repeats, and in case everything goes wrong, you've got your extra copies. So your PCR machine involves this thermocycler machine, and you would need to add your DNA fragment that you want to be amplified. You need to add the enzyme DNA polymerase, a particular type called Taq polymerase, primers, and DNA nucleotides. So Taq polymerase, this is a version of DNA polymerase that is taken from bacteria that are extremophiles, meaning they can survive in very, very high temperatures like hot springs. And as a result, they've evolved to have a version of DNA polymerase, Taq polymerase, that has an optimum temperature far higher. So we use this so that we can increase the temperature very high in this machine so the reaction happens even faster, but the enzyme doesn't denature. Primers, these are short sequences of DNA bases that are single-stranded, and they are needed to enable DNA polymerase to attach. So that's what we need to add into it. And then the process is, initially the temperature is increased to 95 degrees C. Now this is done so that the DNA fragment that you've added is going to have the hydrogen bonds break and it separates into two single strands. Then we decrease the temperature to 55 degrees C. We need to do that so that hydrogen bonds can form, because our DNA primer, which is that short sequence of DNA bases complementary to our sequence on our DNA fragment of interest, those are going to be attracted and aligned, but we need the temperature to be dropped so that hydrogen bonds can form between those two. Then we will increase the temperature so that the enzyme DNA polymerase can then align all of these DNA nucleotides that are going to be attracted to their complementary bases. The DNA polymerase is then going to catalyze the condensation reaction, forming all of those phosphodiester bonds between the adjacent nucleotides. And that is at 72 degrees C. You do need to know those temperatures as well as this process. And that process will then happen on repeat. So you can end up with a huge quantity of DNA very rapidly. And this goes back to that maths formula that you can use. You would do 2 to the power of n, 2 meaning every time it doubles. N is the number of rounds of division that you allowed. So you could work out how many DNA molecules you'd have at the end. And if you didn't start with one DNA molecule, however many you started with, you would multiply by. So if we started with 100 fragments, we would then do 100 times 2 to the power of n. And if you left that going, let's say for 10 rounds, we would then do 2 to the power of 10 times 100, and that would tell you how many DNA fragments you'd have at the end. So the advantages are, it is automated, so it's more efficient, it's rapid, you can make 100 billion copies within hours, and it doesn't require living cells. That means it's quicker and less complex techniques are needed. So a few key terms as well that you need to be aware of. DNA probes, so this came up in the exams this year. They needed to define what a DNA probe is, and a lot of students struggled. A DNA probe is a short, single-stranded piece of DNA that is complementary to a specific allele or gene. Those probes are also labelled, usually with radioactive isotope or fluorescent marker, to allow detection. The process of DNA hybridization, this occurs when a DNA probe is going to bind to a complementary sequence of bases on a DNA sample. And to prepare the sample, the DNA is first extracted and heated so that it is separate strands. Then the probe is added so that it can bind and hybridize. And the point of doing this is you can then use the presence of that probe on a sequence of DNA to identify a target allele. And that's because if you've labeled it with a radioactive isotope, when you use X-ray film, it will be visualized, or if it's a fluorescent marker, it will visualize in UV light. So it's used, these DNA probes are used in medical genetics to screen individuals to see if they have particular alleles linked to inherited conditions such as cystic fibrosis or Huntington's disease, or the gene here linked to breast cancer. They are also used to identify variations in genes that affect drug response, allowing doctors to predict how a patient might react to certain medicines. And this kind of screening can help to detect individuals at increased risk of disease even before the symptoms occur, which allows for early intervention, monitoring, or even preventative treatment. So this is what we mean by personalised medicine and genetic counselling. We're screening for the presence of that particular allele, selecting personalised medicines based on your genetics. And that's because some drugs are more or less effective depending on your genetics. And that enables it to be more effective to you, but also more cost effective as a treatment. And genetic counselling is a type of social work giving people advice and information following their screening of disease-causing alleles. So then we go on to how this process might occur using gel electrophoresis. And VNTRs are needed for this. VNTRs are variable number tandem repeats, and an organism's genome contains many regions of non-coding DNA, which are VNTRs. So they're short sequences of DNA bases repeated a variable number of times among individuals. And the number and length of the VNTRs is very different from person to person. And that is very useful in DNA analysis because VNTR patterns are inherited and highly specific. So the probability of two unrelated individuals having the same VNTRs is extremely low. So we can use VNTRs to examine relationships in terms of genetic similarities. Genetic fingerprinting is the technique used to analyse the DNA fragments containing VNTRs. And the process starts with the amplification of DNA using PCR so that you've got a large sample of DNA. The DNA is then cut into fragments using restriction enzymes, separating the DNA at specific sequences. And those fragments are then separated using gel electrophoresis, which will separate them according to their size, so the number of bases in the VNTRs. A DNA probe is then added, which binds to specific VNTRs, and the pattern of the bands forms a genetic fingerprint. And this pattern is unique to an individual, except for identical twins. So let's go through that whole process in more detail. Genetic fingerprinting, so first of all, you would collect your DNA sample. And the smallest sample of DNA can be collected, and that's because we're going to use PCR to amplify it afterwards. So any cell that contains a nucleus can be used. Then we have the digestion stage once we've collected and extracted the DNA. And the digestion stage is when we use restriction endonucleases to cut that DNA up at the recognition sequence or restriction sites. And enzymes which cut close to your target VNTRs will be added, and we get that palindromic sequence with those sticky ends. Now we need to separate out all of those cut up VNTRs, and the DNA samples are loaded into these tiny wells in agar gel. The gel is then placed in a dish which contains this buffered liquid, and an electrical voltage is applied. The DNA, because of that negative charge on the phosphate group, is going to be attracted to the positive end, and therefore the DNA is going to move towards this positive end through the gel. And the reason that it moves is, first of all, DNA is negative and it's attracted to the positive end, but it moves different distances because of how many DNA bases there are. The bigger a DNA base sequence, the heavier it is, so it's not going to move as fast or as far. If it's a shorter piece of DNA, fewer DNA bases, and that means it's lighter, so it can move through the gel much faster and it'll get further along. So that stage is gel electrophoresis, and that agar gel creates the resistance, and this is what I was saying about the smaller pieces can move faster and further. And that is how different lengths of DNA or VNTRs are separated. We then add an alkaline to make all of that DNA single-stranded. Then we can add our DNA probes to align to those single strands. So we said the DNA probes, those short single-stranded pieces of DNA, complementary in base sequence of the VNTRs. Those probes will be radioactively or fluorescently labelled. So then if they hybridize, we then rinse it afterwards, and then the agar gel, this bit you often don't get assessed on, but the agar gel will eventually dry out and crack. So we do actually transfer it usually to another material that is longer lasting, and that can then be used to visualize your results. And here's one that I did over 10 years ago now, which is absolutely mad. I can't believe that was 10 years ago, but this is looking at the different VNTRs in bacteria, and we could then analyze it to see, here are our knowns, here's my unknown, and I could see my unknown had the same DNA bands as bacteria number 3. So I knew my unknown bacterial sample was the same species as bacteria number 3. So that can be used to interpret paternity tests as well. So we could look at the mother, the child, and any band that the child has that isn't from the mother must be from the father. And that's how we can use it to work out which person must be the father. And I think actually in this one, there aren't any that are a perfect match. Number 1 almost is, but there's this band here that isn't in number 1. So the uses of this genetic fingerprinting could be paternity testing. It could also be forensic science to identify if someone has their DNA at a crime scene, to place them at the crime scene. Medical diagnosis to see if you have particular alleles linked to inherited diseases to help with early diagnosis. Animal and plant breeding to make sure that you aren't about to breed 2 individuals that are very closely related. And also assessing genetic diversity to look at how much genetic diversity there is in a population. So that takes us to the end of topic 8. Hopefully you found it helpful, and if you did, don't forget to scan here for my mark scheme specific notes, which also have recall questions, examiner tips, summaries, key marking points, and essay links. Or my flashcards, scan here, where you can get mark scheme specific key points. But that is it for today's video. Hope you found it helpful, and I'll see you in a video very soon.